What's up, everyone? It's your boy JASHABRAHM. Oh, that was an M. We fucking back. We back. Oh, today was a really weird day. Not gonna lie, it was so less unproductive. I remember sleeping after the math class. I just came back, bam, slept. But yo, that's pretty fast. How you doing? That is pretty fast. Not gonna lie. I did not. I think that's one of the fastest. Let me just adjust the camera a little bit. I feel like that's one of the fastest chat I received on the stream. Damn. How you doing, brother? How was the classes and shit? I believe the latency is much better, right? When you type something on the chat, like it should be better than yesterday. That's what I understand. It's been good, and you. I gotta say, I should have started studying a bit early today, but I don't know. Maybe yesterday's whole sleep schedule went wrong. Maybe I slept over to today in the morning. Yeah, much better. Oh, the latency. Yeah, I changed it now. Yeah. So what I was saying. Yeah, I remember after coming back from math class, I slept over. Then I was just on my bed for a long time. Fuck. That's not good. But at least we back. So right now, today, I should be working on my bioinformatics self-study. I'm trying to understand what are the tools that we have as part of bioinformatics. What sort of tools do we have that help us understand these these molecules, biomolecules, DNA type shit? So I remember the last thing I did was Kamer analysis. Oh, okay. Well, well, I gotta check my stream because I believe there's in the description Restream shamelessly plugs in their website. Restream, stop doing that. Yeah. And I missed the deadline for protocol submission, one of the protocol submissions. So I gotta send an email to the professor or the invigilator on this. Please don't strike me down. I will do it ASAP. Type a message. $15. Okay, so where do I start? Should I send the email first? I think that would be the better move. I send the email first. Yeah. Yeah, for let me think. What did I say right before this? What did I say? Send it. No, I did say something before sending the email. Before me saying send an email. What did I say? What did I say that makes you send a yeah? The email. Oh, okay, okay, okay. I thought I slipped in something else. Okay. So, GPT, could you help me with the plug this out? Yeah, there you go. Well, where's my email preparation chat? Email drafting, there we go. Patience. Patience. Where's... I should start looking into LinkedIn. But I cannot do everything at once, right? No, I can't. If I remember, the name of the invigilator is Lucas. Lucas. Fuck. Damn, the laptop screen is restrictive. Lucas? Individual protocol discussion. No, that's not what I wanted. Fuck, I should have submitted it yesterday. Yesterday. With section 2. Which pages do I have to rewrite? Ten minutes? Okay, come on, fast. So I gotta do first page, repetition. What? Stupid. Damn. Fuck. Okay, I gotta write it on the next page. What else? The structure. I have to draw the H2O structure. Okay. And the calculation of yield. That's where I messed up. That's where I messed up. Okay. Draw the apparatus. Okay. So basically, I have to rewrite the whole protocol. Log in, bro. Where is his email? Can I get an email, please? Can I get an email, please? Please, can I get an email, please? Can I get an email, please? Where's the email? Lucas L. Oh, come on. Yeah, there we go. Teacher. Okay, so they are not giving me the email. I gotta search on Google. I did see something. No. Okay, I can't. Okay, I don't think this teacher has their email publicly available. Yep, there is, but okay, I got the email. Please forgive me. Okay. I want you to prepare me an email regarding my correction and resubmission of my general chem lab protocol section 2, as I have accidentally missed the deadline for the corrected submission of the corrected protocol, right? That's what they say. For the section until 10:11, please hand in the original version plus correction. Dude, some of these rap songs are so bad. They really be yapping. Oh, okay, there we go. There's the email. Please review all the corrections. Okay. For submission, one, and I apologize for the inconvenience. Can I submit it within tomorrow? I could bring it to your... Should I write that? I could bring it to your office. Fortune match. I am with my lab to Yeah, I think that's okay. It's okay. I don't know. Like submitting the protocol inverse. Too much. Should I write that? $15 is already... Okay. I have written what I have understood on K-mer analysis on my flowchart, but I have only written the basic idea on what K-mer analysis is, but I haven't specified what I have done using K-mer analysis. Where's GPT? No, can I get a can of Coke? Yo, average Sans fan, how you doing? And hi K, hi K. What's going on Discord? Nothing's going on Discord. No, something's going on Discord. Come on, just open it. Just open. Please a copy. Then I paste it. What? Studying for a test right now. Yo, let's go. Let's go. Now that's something relatable. Which type of test? Is it school? I mean, it's gonna be school related, right? Let me know what the which type which chemical equations and stuff. Basically atoms. Let's go. Let's go. Uh, so if you don't mind, I don't if I don't know if you don't mind me asking, do you know what exactly you're learning right now? Like what's open in front of you right now? Is it chemical equations or is it like atomic orbitals? What else is there? Types of bonds? Balancing chemical equations. Let's go. Now that's one annoying, not annoying, it's just what are main parts of balancing chemical equation? There's the oxidation half reaction, there's the reduction half reaction, and you gotta find tedious. So you're saying it's tedious or tedious? That means hard, right? Please tell me it's hard. From my understanding, the meaning of the word tedious is hard, or it's more work, something that is boring, tiring. Okay, so it's not hard exactly. Okay. Uh, basically, yeah. Okay, yeah. That's the balancing part. You got. You also have to look into the oxidation numbers, right? Before you balance. Like how you figure out if amount of electrons are equal on both sides. You look into the oxidation state, right? I'm also naming compounds. Yep, that's also fucking it's not hard. It's just uh, you gotta learn some common names, like phenol, not phenol. Yeah, there are ring structures. When it comes to ring structures, it's gonna have a different name. But yeah, once you start figuring it out, you feel really intelligent when you start naming compounds. Maybe it's just me, but yeah, balancing chemical equations. It is kind of some a little bit math to do, but not too much. Also, if I understand, you should also know some oxidation states, right? Like oxygen has an oxidation state of -2, hydrogen has +1, carbon. Fuck, I don't remember what's one for carbon. Damn. Common oxidation state. +4. Okay. So is this test based on school? Is it the school exam? Yeah, school. Well, good luck, brother. I just gotta say, the more you learn, the interesting it gets. Not gonna lie. Damn, I feel bad for doing this. I feel bad for late submission. Accidentally missed? Yeah, accidentally, because I just found out today that the submission day was. Oh, come on, please. Because you're mixing, that's because you're mixing general concept versus your implementation. You need both. First layer, what K-mer analysis is. I know what K-mer analysis is. At least I have a basic understanding on what that is. Now I need to know what I did specifically. Where's PyCharm? You can spread the little. So I looked into these sequences, specific sequences. Sans fan, if you, I don't know if you're still here, but how are you studying? Like, do you have textbooks to refer or notes, or just straight from the internet or from stupid ChatGPT, something like that? How is your how's your form? How's your form of education? How do you what's your source? Not form, source. Where is it? So I have wrote till enrichment tells us slide the 6 window length across the entire genome, count every possible 6-letter DNA pattern, Enrichment tells us certain sequences biologically favor, as 4060 are possible in which 4 raised to 6 ATGC, total number of 6 mers in genomes, divided by every possible 6 mer could appear this many times. So if I take that value x, expected frequency under randomness. Now, number of specific sequences, example AATTCC. Which one was AATTCC? Okay, I did not even go for that. Okay. Divided by the expected frequency under randomness, which gives a percentage. And if the percentage is greater than 1, that means that sequence is biologically favored. If it's equal to 1%, biologically random. If it's less than 1%, it's biologically less favored. So that's what enrichment as part of K-mer analysis does. Now I need to understand what I specifically did. And these sequences are mostly seen across all living organism in general? Question mark. $10. Not exactly. This is an important distinction. Those sequences are not universal sequences seen in all organisms. They're more like known biological motifs commonly associated with certain functions. Some are widespread across life, some are mostly eukaryotic, some are mostly plant related, and some are artificial lab related sequences. Very common in eukaryotes. Transcription factor binding motifs very important in plants associated with light responses, threats, growth regulation. This one is biologically meaningful for your bamboo work. Bamboo work? Yo, my eye is twitching. Okay, can I get this as a para explaining what are the sequences I have used? What are the sequences? Sequences. What are the and what each sequence is for? Whoa. The target sequences used in the camera analysis are non-biologically important DNA motifs. Okay, this looks more para. I would like to have as bullet points with a small in a single sentence what those so oh come on, give me for the whole thing. Yep. So that's Kamer analysis. I gotta change how come on, I gotta use my laptop just for this. The laptop trackpad. So under your camera section, bus frequency distribution of 6 months. 10 minutes, okay. 15 minutes. Fuck, I missed the section 2 date. So stupid. Do I have the results? Yeah, there we go. No, that's not the result. That's a Python file. Come on. It's not the result. That's not the result. Wait. So there's no result. We'll just. Certain regulatory motives appeared enriched relative to random. Okay, wait. Rerun it. Capture only the biological meaningful outputs. Oh, fuck. I'm gonna move the laptop towards me a bit, because, yeah. If this is gonna get me it's doing the right analysis, but your current script has only one major gap. What your code will successfully give: genome wide k-mer statistics, total fixed-mer count, unique k-mers, enrichment signals for known motives, biological interpretation clues. This is what I actually want for promoter like signals, regulatory signals, translation trick signals, epigenetic patterns. This is what goes into your overall. Okay, let me just run this. I feel like I should have saved it, right? I should have saved it somewhere. Why is it not saved anywhere? Results from my code. 6mers.pdf. Got him. Got the motherfucker. Top 20. Top 20 most frequent 6mers. Biologically important k-mers. So we got the start signal. That's pretty relevant here. BL12 restriction site. Eco R1 restriction site. That's one. That is biologically random. So very less interpretations. Damn, it's taking a lot of time. Not gonna lie. But biology just means low complexity or repeat rich DNA. But interpret it correctly. Genome-wide averaging. You scan the entire genome, promoters plus introns plus repeats plus junk DNA all mixed. Signals get diluted. K-mer is very short signal. 6-mers are very tiny, too short to uniquely identify genes, so they appear everywhere by chance. Your motifs are not guaranteed to exist in bamboos. Some motifs are plant conserved. What should you conclude? This is your reveal that most biologically known. Come on, just let me read it, bro. Let me read it. All right. Oh yeah. Biologically known motifs show weak enrichment compared to random expectations, while repetitive sequences dominate the genome. This suggests that the global motif frequency alone is insufficient for gene identification, and positional motif scanning is required for functional positional motif scanning is required for functional interference. So how do I proceed with positional motif scanning? What do your result actually means in GT? What this tells you about structural noise such as repeats, target motifs, weak biological signals. Genome-wide statistical bias, not gene-level signals. This is what exactly knocks motif scanner, your next stage. So positional motifs. So 15 minutes. Positional motif scanning is what the knocks finding where a motif occurs in genome, exact coordinates. not just counting it. So instead of so tomorrow I gotta lock in. Fuck, the lights is fucking annoying, not gonna lie. Ugh. Ugh. Guess who's back? K, what's up? I'm guessing you're back from something important, or was were you playing games or some sort? With your Knox motive scanner, but one important clarification: what positional motive scanner actually means. You know what? I want to add my result onto this PDF, onto this workflow. How do I add it? I have it here. Can I add it just like that? Oh, I can. Okay. What? I was just playing with my cat. Oh, okay. Uh-huh. Yo, okay, I gotta say, tomorrow it's gonna be something special. It's gonna happen. We're gonna play Sleeping Dogs tomorrow at 8:30 p.m. Hell yeah. You know what? Just so I said that, I need to set that stream. I'm gonna watch that. If you could, that would be cool, not gonna lie. That would be cool. But you know what? Let me just set that, let me schedule that stream. Because, yeah. I feel if it's scheduled, it's gonna be useful, more useful. Schedule stream. Create new. So it should be part 5 of Sleeping Dogs. This would be the part 5, and we are progressing on the story. Not too much, but 30 minutes is not going to be too much progress, but not going to lie, playing video games once a week. I got to study biology for tomorrow. I asked my classmates and they don't have it. What? You have an exam tomorrow and you and your classmates don't know what to study. Whoa. Like, you could contact your teachers? How about that? Are they gonna be like, no, you should have known better? Are they gonna respond like that? We know, but I missed the lesson, and my classmate just like didn't write the chapter like with a friend and has it, but doesn't want to send. Yo, yo, that's stupid. I remember there are some students who just won't send whatever happened on the class, or some students just don't send their notes. On one side, I heard from, like, I told this, I had a conversation with a friend who does this, and he told me, like, I worked my ass off for this class while you were just back at home or something, and why should I just give you my uh, work even though you did not do nothing for it? I kind of see the point where students don't give out some stuff like that, but come on, it's not gonna affect you, is it? No, I don't think so. Like, if it's gonna take too much of your time, maybe okay, but sending the notes, I don't know. Do you know what, at least what which part of biology is it gonna be? Damn it, it's been a long ass time since I learned biology. It's been a long time. Oh, what do we used to learn in biology? Oh, in class 12th, I remember. Yeah, I know. Okay, can you tell me some chapters or some topics? So you do have biology. I thought you did not have biology as part of your college or school. Oh, okay. Yeah, okay. Do you remember any, or can you tell me any topics that is gonna come for tomorrow's exam? Evolution. Oh, evolution. Changes of species over time. Successive changes of species over time. Oh, okay, okay. Evolution. The phenotype, dominant gene, recessive gene. There is a cross box. Ah, fuck. God damn. Global frequency information, positional motive scanning is required to. The global multi-frequency compared to random expectations on while repetitive sequences dominate the genome. This suggests that global motif sequences is alone insufficient for gene identification and positional motif scanning is required for functional interference. But normally I think it's a document and I'll need to read it. Hopefully that shouldn't be too hard. Okay, I'm guessing you're planning to read it tomorrow morning? Or are you reading it right now? What was that? No, like there will be documents and I'll need to answer questions in the exam. Wait, they will give you the document during exam. What? Did I understand that correct? It's like an open book? What? That's cool. If it is, that's cool. You could read this document during exam. It's not good due to sheer gDNA size and yeah, it's usually like this. Like the exam, there will be documents about things and I'll need to answer questions about these. So, is it like a final exam or is it like a midterm? How is this? Or is it like a random test? Is it? I don't think it's gonna be a final exam because it's May. May. Random test. Oh, okay. So it's not gonna affect your final grade, right? Or do they consider all these random tests for final grade? And this results in a lot of waste or all these randoms counts high. Oh, okay. Oh, they do consider all the tests, okay. Well, I hope it's gonna be okay, right? $15. Figaro, Figaro, Figaro. Is it the lights that's causing me a slight headache? It's a very slight headache, but it is there. Waste or noise, which destroys the information that we need, and as part of that we are going for motive scanner. But my another would be, is there any way to split the DNA into smaller chunks and observe? Would that help in figuring out how the target occur? You're splitting, yes, splitting the genome into smaller chunks is standard and useful idea, but it only helps if you're clear about what problems you're trying to solve. First correction, Kramer analysis is not bad. Your interpretation is slightly off. It is failing because it's positionless. Tells you what patterns exist, how often they appear, but not where they are, whether they form genes, whether they're functional. It's not noise. It's just low resolution information. Okay, you know what chunking the DNA genome actually does is split the genome into windows. You got the window-based Kramer analysis instead of global counts. Local frequency gets per region, regional enrichment references, hotspot detection. What this helps with, chunking helps you detect regulatory hotspots, regions where motif clusters more than expected. Gene rich versus gene poor regions. Some genomes regions are dense in functional signals. Local motif enrichment, example T. That is biologically meaningful. What chunking does not solve, it does not give you exact gene boundaries, exon intron structure, through gene function, because even small windows still lack biological annotation context. Why your pipeline moved beyond Kmers? Your current logic is correct. Stop bullshitting. So chunking is an intermediate upgrade, not the final solution. Okay, so I could look into that. Okay, so that means, well, so that means I should consider the chunking part also. Hey, this is not an assignment. I'm looking into various ways I can understand. So this is also an option to try out. So I'll be adding onto my workflow. I'll explore this later, or there's some good movies coming up. Oh shit, I gotta schedule the stream. Schedule. I need to have a thumbnail. 13th of May, the 2030. There we go. The stream is scheduled, but I need to have a thumbnail for the stream. And if I need to have a thumbnail, I gotta make a thumbnail, which is gonna take some time, which I don't have a lot. I'm saying time is not that easy. Mm-hmm. $12. We're in the genome motifs clusters. Important limitation: does not define gene, does not define regulatory logic, improve motive correctness. It only adds spatial resolution, not biological interpretation. I have for more. Oh, what was that term? It was a good term. What was the term? Spatial resolution. I could. Okay, now I got an image. Now I need to add it to just paste, bro. Come on. Fuck, just paste. Oh, we got the results. I mean, I didn't need the results though. I already had it. Already had it. Why is it not found though? Camera analysis of a person, you know. Fuck, that's too big though. What? Where is the file though? Oh, okay, got it. Kim. There we go. Okay. Oh, it's not working though. I forgot how to erase. So I'm trying to make a thumbnail and I'm still figuring out how to erase shit. How do you erase? Okay, it's erasing, right? It is erasing, but it's too small. How do I make it bigger? Okay, there we go. There we go. I just want my face up there. I need a bigger screen now. Just a minute. Whoa, what happened there? 10 minutes. What happened there? Why is it looking like this now? I don't get it. I don't get it that far. What the fuck happened here? Why is it only halfway through? I don't get it. I'm gonna close it. Discard. I just need that this is not the way to go, as it's only giving us a very rough picture. Oh, I mean, that's what it is, right? It's only giving us a very rough picture. motive scanner. $16. Okay, so it's now time for motive scanning. How do I write that? No, oh, yo, K, what's up? I don't have to say what's up, but yeah. Yo, I was actually in the middle of preparing a thumbnail. I could show you how I make my stupid ass thumbnails. So right, let's just keep this out of the way for a bit. Now I need to add my face instead of this guy Way's face. How do I add it? I I have a I have a picture of myself. Hell yeah. Hell yeah, brother. All power to you, brother. Now how do I change uh image? Fuck, I forgot there was a tool. Transform tool, crop. God damn. God damn. Hell yeah. So I forgot how to. Oh no, 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 that's not. It's not how it is. That's not what I want. Let's now. Okay. No. How do I get my face only? For everything, let me first erase. Yep, erase all my unnecessary parts. I just need my face. That's where I gotta be careful. Oh, I can't even see my ear. Oh, the sensitivity is too much. The sensitivity is too much. Okay. Just a little bit. Just a little bit. A little bit. I don't even know how my face looks at that side. It's too dark to see. But it doesn't matter. Don't matter. Yeah, whatever. How do I transform? I need to shrink it. Scale? I think that's it. No. There we go. Got it, got it, got it, got it, got it. Where is it? Fuck, I can't. Yeah, 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 there we go. Yeah. Works. That works. Yep. Well, I guess that's it. Sable. 10 minutes. It's 10 o'clock. File. Save as. Export to downloads. Yeah, there we go. No, not pick, just downloads. Yeah. GPT is too fucking slow. This step shifts your pipeline from statistical DNA analysis to genome localization of regulatory signals. Motive scanning equals locating exact DNA pattern in genomic coordinates. What are you doing now conceptually? Instead of asking plain text, how often does TATAA appear? Uh-huh. Where is the genome that tata occur and what is near it? What this step produces? Your motive scanner outputs chromosome ID, position index, motive identity. Okay, now the important thing is, I don't think I should continue with this now. I should continue with my timetable. Because it's been a while since I done timetables. I think. Timetables. Denk man. Also ich hab auf Deutsch lernen, ja. Das auch. Oh, wie soll ich diese Zeit vermachen? Wie soll ich das machen? Äh, morgen habe ich Biochemical Thermodynamics kurz, habe ich Thermodynamics, dann habe ich Organic Chemistry, ich muss dieses submitten. Okay, I have decided that I should prepare a timetable, but the thing is, I feel like I need to make that Reddit video, but do I have to focus on shit like that now? Is there any better subreddits? Oh, bro, ads. I just wanna ask something, and it's not related to this, but how will R Studio or the R language be implemented in my case? Can I ist doch fair. Sie haben eine gespaltene Persönlichkeiten, das macht 106 Euro. Hier sind 80. Holen Sie sich den Rest von Undertale. Okay. Pass mal auf die Klugschliebser. Okay, gang. Okay, YouTube. I'm gonna end the stream, and I'm gonna look into Deutsche memes. That's gonna be cool. Let me see if that works. Peace out, YouTube. K. I don't know if you're still here, but I appreciate you for checking out. Sans fan, always. Peace.